sensory motor systems, and I was given some leeway on what actually we would cover in this lecture, and I realized a lot of you probably come from more engineering, bioengineering backgrounds, and less neuroscience, so perhaps some of you don't have a great understanding of how the brain itself is organized. Is that the case? Yes. So I remember being in your shoes when I started my graduate work as a neuroscience student, having coming from engineering, and it's really important to have sort of, from an engineering point of view, some sort of logic around which you can understand how the brain is organized. So that's what I'm going to try to convey to you today. And excuse my voice when I come back in a month or so. Hopefully I won't sound like this, but I had really bad laryngitis over the weekend, so at least I can talk today. Okay. So just to give you sort of a general overview, so what is a sensory motor system? Sensory motor system is just this idea that when you're in your work, the world that you're in every day, you basically generate motor behavior in order to interact with the sensory stimuli that you're surrounded by and act on those as you decide, right? And you can think of everything from reaching and grasping the coffee cup, which I usually have next to me, I don't right now, and what's involved in reaching and grasping the coffee cup. You have to sort of see the cup, figure out how far away it is from you, get your hand ready. You actually use proprioceptive feedback from your hand to shape your hand properly. There are a number of things that you're doing as you do this move that are integrating visual, proprioceptive, and ultimately tactile inputs, right, in order to accomplish a pretty simple task. Same as if you're just walking through space. In order to walk through space and not fall over, you have to integrate multiple modalities. So if you know the textbook Candell, Schwartz, and Jessel, Principles of Neuroscience, most of you. So I borrowed some slides from that textbook and another textbook, which is Paradiso and Connor, which is a group of people at Brown to talk about the development part. So a lot of the figures are from these two textbooks. You can always go back to those chapters if you want. I'll post what those two chapters are. Okay. So you can consider an even more complicated behavior like returning a tennis serve. And first, in order to return the ball, you have to actually judge the direction of the ball, its velocity. You need to also sort out how fast you're moving towards the ball. So use your vestibular system, your proprioceptive system. So you're using multiple modalities just to get, to perceive how and where you are. Then you need to use premotor cortex to plan the actual movement you're going to generate. Are you going to do a forehand, a backhand? Got some sort of executive decisions to make. And if you're just been studying all day, you might not be as motivated as you might be if you're doing this first thing on a Saturday morning, right? So you've got your amygdala, hypothalamus for motivation, and then of course you've got to get the action part going where you gracefully complete this backhand or forehand and have everything coordinated. And so it's a multi-step process. And in order to do this, you're using many parts of the brain. So visual cortex, other sensory cortices, premotor cortex, again, these areas deep in the brain, like hypothalamus and amygdala, and then motor cortex. And these are sort of shown schematically here, right? And it's really just to give you an overview, so you don't have to kind of cram this, but just to give you a sense that to do one behavior, you're using the brain sort of in concert. Okay, so what I'm going to do today, as you go through these different parts of this class, people are going to be talking about their favorite structures. So Shashay Wang will talk about the auditory system, and Reza may talk about premotor cortex. So we'll have different people talking about different areas. The question is, how do they all fit together in the big scheme of things, so you're not just memorizing a whole bunch of different words, right? You really can map them onto some logical structure. So what I'm going to start with is what I call the brain's basic building blocks. And the idea is that there's a functional organization of the brain, which if you kind of carry this map around in your head, it's actually easy, relatively easy, to keep track of things and where they fit into this sort of bigger picture. Okay, so these are a couple figures that are taken from the Candell, Schwartz, and Jessel book. And they show you, if you look at a mouse brain, right? Or rat brain, I'm not sure if that's a mouse or a rat. It's a very linear structure, and it's actually easier to understand how things are organized if you look at a brain in an animal like a mouse or a rat. Then if you look at the human brain, it looks much more complex, right? It's all this stuff going on. You've got this big cortical part, you've got the cerebellum off here, then you have this brainstem that comes out at kind of a right angle. But really they're very similar, and what we'll do is go through why they're so similar and help you better understand what you're looking at. So, just backtrack here, not backpack. So if you look at this first panel, you can think about the way that you're looking at a brain slice, you want to try to understand how you're looking at it, right? What are the, what's top, what's bottom, what's left, what's right. So people talk about dorsal, ventral in panel A. So dorsal would be towards the sky, ventral would be towards the floor. Okay. And then you have rostral and caudal. Rostral is like for nose in Latin, so it's towards the nose, and then caudal is towards the tail, for tail. Right, so those are two coordinate systems that are right angles, right? Up, down, forward, backwards. And then you have an additional one, which of course would be medial lateral, right? So between those three axes, that's generally how people look at the brain and try to understand how it's cut. But what you can see right away is if you do those same cuts in a mouse brain or rat brain versus a human brain, it's going to be quite different because this part of the structure, which is running sort of rostral caudal in a rat, is actually running, right, dorsal ventral in a person. And that's what gets a little confusing when you start to look at slices from people doing MRIs and things like that. Okay. And then it's, panel C just shows corresponding reference images that you use for brain slices when people do imaging. So you've got this, basically this horizontal slice, which would be running parallel to the floor. You have a coronal slice, which is running this way, and then the sagittal slice, which is just sort of cutting down through. So generally, if you're looking at it, like the inside parts of the brain, you might look at a mid-sagittal slice, and you can look across multiple structures on the inside rather than outside. Okay, so this is the point I just made in the previous slide. So what you see here in this rectangular box, right, is a part of the brain that in a rat or a mouse is actually running parallel to the floor, right? So in humans, it's actually rotated 90 degrees. And this part of the brain is usually called the brain stem, but also has some other parts like the thalamus up here. And we're going to go through each of these subdivisions in the next slides. Okay, so in trying to understand the basic blueprint of the brain, there are a couple different approaches you can use. So one is developmental, and that's what I'm going to start with. You can also look across species, because as you already noticed, a mouse brain looks a bit like sort of a brain from a human that just doesn't have all this extra cortex. So what we're going to just do is go through an analysis of the sort of development. There's some terms here that you need to know, but in the end I think it's useful because it'll give you really a blueprint of how the brain is organized. So if you look at the neural tube, which in an embryo is the beginning of the nervous system, right? You basically have these three subdivisions. So we're looking basically top down on the embryo. Most rostral towards the nose, you have something that's called the proencephalon. And then just behind that, you have the mesencephalon, and then behind that, the rhombencephalon. And those are sort of also commonly termed, respectively, the forebrain, the midbrain, and the hindbrain. Okay, and those are the basic three building blocks, which all brains come from. So as development sort of continues on, what you see, particularly for, say, a here human, is this huge expansion of the prosencephalon, where you end up with this huge sort of development of what are called the telencephalic vesicles. Within here, right, still along what was the neural tube, you now develop what's called the diencephalon. That will become the thalamus. And then again, we have the mesencephalon and the rhombencephalon back here with the midbrain and the hindbrain. Okay. So again, as development continues, you have this forebrain expansion where you end up with these paired telencephalic vesicles, which you might guess become the cerebral cortex that you see on the human brain that's so big. You have these paired optic vesicles here. Does anyone guess what those might be ultimately? That's the retina. That's actually what the retina is. So you have basically the nerves will go out to where your eyes are, and those cups are what will be your retina. And then you have the diencephalon, which is the thalamus in the middle. Okay. Okay, so again, major sort of landmark in evolution is this development of the forebrain, so it gets bigger and bigger. The cerebral hemispheres swell and grow posteriorly, so they get bigger and bigger, and then eventually they start to wrap around the rest of the brain in what's called sort of a ram's horn. And when you look at a human brain, that's why it looks so complicated, right? Because you basically have this wrapping around of these telencephalic vesicles. And again, the olfactory lobes, which we saw back here, is, well, I guess they might do that. That you have at the very front end here are very variable across species. So in humans, the olfactory lobes are not very big. They sprout off the tip of each of these telencephalic vesicles. In animals like dogs, relative to the rest of the vesicle, they're huge, right? So we're actually a little smell, constrained compared to other animals when you look at the total, I guess, dimensions of our olfactory lobes versus those of other animals. Okay. So then what you can do, so this is just all looking from the surface, is you can start to make slices through the brain, right? So you can really understand what's the organization within it. Let me just scoot back here. What I should note is this neural tube is ultimately all filled with fluid. So there's like a fluid core that runs through the middle of it, sort of like there is in the spinal cord. I should also note down here, after the hindbrain is turned into the spinal cord, so it's all one continuous structure. So if we make a slice through the forebrain, right, or the telencephalon, at the most rostral part, you would see basically these two spaces here and some space down here. And that would all be filled with cerebral spinal fluid. And in fact, there's a channel of cerebral spinal fluid that runs from the telencephalon back down through the mesencephalon, rhomencephalon, and down to the spinal cord. It's all continuous. So when you're looking at slices, you'll, sort of a hallmark of being in the cerebral cortex are these ventricles, right, that are filled with cerebral spinal fluid. So this would be here, the cerebral cortex up on top. Down here in the middle, right, is that diencephalon, basically the thalamus that the two lobes have wrapped around. And then here you have what was this remnant of this channel that's now these two ventricles that are called the lateral ventricles up in the front part of these lobes, right? So you have the lateral ventricles, and then the third ventricle is down here, right over the thalamus. Okay, and I'll show you another cutaway that shows you this ventricular system so you get a better sense of how it all fits together. Okay, so basically these are coronal sections, right? And they show here are the ventricles, right? And remember the ventricles are the fluid, the gray matter, right, in C, and then white matter. And I should just note, so gray matter would be neurons, right? Neurons, cell bodies, and gray matter would be projections. So the way that this is set up, labeling here shows you where the ventricles are, here where a bunch of the different nuclei and cell bodies are, and they have different names where they sit in different groups together. And then down here in D, you can see some of the structures which are big axon tracts connecting one nucleus to another, or parts of cortex, getting input from certain areas that are very large tracts are labeled here. So for example, just to be clear, like the corpus callosum, which people have probably heard about, that connects the two hemispheres, is something you can see pretty much by eye. It's such a big tract. Okay. So we focused so far on the development of the telencephalon, and now if we go, or the prosencephalon. Now if we go back to the mesencephalon, what you see here, right? Initially you have this sort of tube that basically develops into something more complex with these two big lobules that wrap around. In terms of the mesencephalon, which is here in red, it basically develops in a far more simple way. It has what's called a roof and a floor. So the roof is the tectum, and the floor is the tegmentum. And those terms are actually used when people are describing structures. So there is a structure that you'll hear about later in the class called the optic tectum. It's also called the superior colliculus. That's just the roof of the mesencephalon. And there's an auditory portion called the inferior colliculus that sits just behind. So that's where those two structures are. So you can cut through this, and as it develops, you see the tectum up here, you see the tegmentum down here, and again, that center canal is filled with cerebral spinal fluid. Okay, so this is a sagittal view showing the basic plan that I already mentioned, right? So we have, what was the prosencephalon, now containing the telencephalon and the diencephalon, right, which is the cortex and the thalamus. Then you have the mesencephalon here, right, which is at the roof and the floor. And then behind here, you've got some other subdivisions in the robincephalon. And the robincephalon basically turns into the cerebellum, right, which is that little sort of cabbage-like thing that's below the cortex. Has the pons and it has the medulla, right? And those are areas that are really important we'll talk about in terms of controlling eye movements you know, the premotor and sort of also breathing in the medulla. So some of the really basic things we do are controlled by nuclei and these structures. Okay, so as I said, you've got the pons and the cerebellum here in an area it's called the metencephalon. You've got the myencephalon, which is the medulla, and these are all parts of that. What was initially the rhombencephalon. And then if we look at a cutaway here, you can see the continuous nature of the ventricular system, right? You can see that the cerebral spinal fluid, which was initially the center of that neural tube, is now has a far more complicated sort of geometry, but is all continuous from structure to structure as it was initially at the beginning of development. So, so I'm now going to flip back to the slides from Canal, Schwartz, and Jessel, and this is pretty much what I've just shown you. Again, we have these sequential stages of neural development, for neural tube development. We have the basic plan, which is this sort of linear neural tube that is then elaborated into ultimately the human brain. And if you go look at other species, actually will look more similar to what it really started to as a neural tube. Here again, we have the optic vesicles, the forebrain, the two telencephalon lobes coming off, and then these other structures. Okay, so there's a lot of work done on trying to understand how you start with a neural tube and then elaborate these very complicated geometries. So there's people are trying to understand and use new techniques, both to visualize individual cells as animals develop, and also understand what are the sort of the guiding molecules that generate this topology that we see. So, I mean, this is really just for your basic interest, but it is a very rich field of research because, as you can guess, when you end up with kids who maybe have autism or, or other types of disorders, the idea is what's happening to their development, and are the circuits organized slightly differently. So, there's a lot of energy put into understanding exactly how the circuitry is set up during development, really understanding how this is normally done, and if there are certain sort of genetic tendencies for things to change, how it might affect the natural progression. So, for example, molecules that set up this topology that are really well known and well studied are for example, called sonic hedgehog, great name. Maybe less exciting bone morphogenic proteins, and then what's called the Wnt family of proteins, right? And you basically have different gradients along the neural tube. So this shows, I don't know how easy it's to see, so this is sort of a beginning development type looking brain, right? So you've got your linear tube, neural tube, and things are just starting to differentiate. And what this shows is, I forgot what kind of fish this is. It's either, does anyone recognize this fish? It might be, maybe it's a goldfish. But if you look down on you look down on the fish brain, it looks, very much like that sort of the beginning of development that you'd see in the human, right? So it's a very good way to understand development, both by looking at different animals along sort of the evolutionary tree, as well as looking at development itself. So just to kind of summarize what I've told you, if you look at a rat brain, or even a fish brain compared to a human brain, there's some really fundamental differences in just looking at them, but the underlying building blocks are really the same. And I think that's gives me hope, right, when you're trying to understand how it's all organized, that you can sort of put it together in a logical framework rather than just memorizing a whole bunch of different terms and kind of sticking them together. So, the similarity, of course, is that overall structure and the basic beginning is pretty much the same, right? But then what you have are differences in particularly the number of convolutions, right? So here, the brain, the telencephalic lobes in animals like mice and rats, even some monkeys, are very smooth, right? But what you really see with more advanced primates, and in particular humans, are what are called sort of these gyri and sulci. So the bumps are the gyri and the indentations are the sulci. And the idea is it's the way that the evolution came up with packing as much cortical structure into our brain cavity as possible. So this is a real differentiator as you go across the evolutionary tree. When you see something that's a really convoluted cortex, it suggests the animal's actually pretty smart. The other is forebrain organization, as already mentioned. Humans really have relatively small olfactory bulbs as compared to some other animals. So if you look at rat, the percent of the rat's telephallic lobe that's olfactory bulb versus the rest is quite markedly larger than it is for us. And again, in humans, what really makes the topology of understanding how the brain is organized complicated is the fact that you've got this sort of ram's horn configuration where the cerebral vesicles both wrap around. And then, of course, the final thing, which is important to remember so you don't get confused looking at brain slices, is that there's a big difference in the brainstem orientation, where if you're looking at slices in one dimension for a mouse versus a human, it's actually quite complicated to look at slices of dorsal ventral cut if you're trying to look at brainstem slices in the human. So again, this is just reminding you of the different ways in which you can look at slices and trying to, I guess, really understanding these so that when you do look at different images or views of the brain during class, you'll have a good understanding of how you're looking at the brain, kind of going back to that idea of the node tube. Okay, so at this point in the lecture, hopefully, or after you've gone back over the notes, you should have an appreciation of the basic building blocks of the brain and the major differences in the brain across different stages of development, the forebrain, brain, and the hindbrain, and the development of this ventricular system. So the ventricular system is different than the vesicles, right? So remember, those are two different things. The vesicles are the prosencephalon, right? The ventricular system is what has the cerebrospinal fluid. And then again, this idea of the difference in the 3D organization, even of the two, rat versus human things. So what I'm going to move on to now is describing, going more to a human brain, which is more like, say, a monkey brain, where a lot of electrophysiological studies have been done, is really understanding the functional organization, and probably going to some of the terms that you guys have heard before, focused more on the different structures and how the brain is organized for basically sensing, sensory inputs, and then generating motor outputs. So what I'm going to do is start with the cerebral hemispheres, which again are what those telencephalic lobes turn into in humans. And if you look at a human brain, again, the main thing you'll see is that sort of ram's horn, which is telencephalic lobe wrapped around. And the cerebral hemispheres basically are characterized by four major lobules, or so it's a little confusing because people use lobe different ways. So you would say four major areas. So we have what's called the frontal lobe up here, a occipital lobe back here. So occipital lobe is really like back at the base of your head. Frontal lobe is up here. You have the temporal lobe, and then you have the parietal lobe. And those are areas you'll hear, like people say, oh, parietal cortex. Parietal cortex is part of cortex in this parietal lobe. And if you think about different sensory systems, they're basically projecting to different parts of the cortex, and I'll show you those in a little bit. You can also look at, there's some major sulci that are landmarks, and people will refer to when you're trying to interpret an image. So the two main ones are the central sulcus, which runs right between the frontal and the parietal lobe. And then there's also the lateral sulcus, which is whenever you draw a brain, that little part you draw. Makes it look like a brain. And then this is a medial view, and what's really clear is, when you look at the outside, oh, and I should note the cerebellum is down here, which is really important for motor control, and we'll talk about more in my lectures. It's involved in keeping your movements calibrated. If you do a midbrain section, what you can see is, of course, we have all the cortex, but here's where you can see all these other parts of the brain. There are actually some deep parts of cortex I'll talk about in a little bit. And in addition, you've got basically the mesencephalon and the rhombencephalon, right? Okay. So if you look at the outside of the cortex, you see these lobes, but what you do miss are some essential structures, and they're really important for both our, generating movement and motivation, things like that. So if we do a coronal section, which would be cut this way through the front of the cortex, right, as it's shown here. So this is a side view. I'm going to cut through parallel to the face. What you can see are there's some other structures, and these are structures people may have heard of the basal ganglion. So they're deep down in here. So for example, here's the basal ganglion. Here's the insular cortex. These are different than the other types of cortex I'm going to talk about. They're not sort of 6 layered cortices. They look more like nuclei. Some of these areas are very important. People here have probably heard of deep brain stimulation. It's used for more and more things. It's used, obviously, for people with Parkinson's. It's actually used for people now who have OCD. It's in people who have an intractable tremor. So it's become a very common approach clinically, more and more so for people with sort of profound chronic neurological ailments. And these electrodes will, for example, for Parkinson's, will go down into parts of the basal ganglion. So they're quite deep, the electrodes that are put down here. The electrode itself is probably, thinner than a piece of spaghetti, just to give you a sense, and they're chronically implanted. Okay, so what I'm going to do now is talk a little bit about neocortex. So again, when you look at all these different, the occipital, parietal, temporal, and frontal lobes, and you look at all these sulci and gyri, if you could unfold everything, what you're looking at is basically a ribbon of this 6-layered neocortex, cerebral cortex. And the neurons of this cortex, you can see if you use what's called different kinds of staining. You have a Golgi stain that'll stain the cell bodies. That's what's shown here. But you can also have a stain, so this would be like a Golgi stain here. Oops. You can also have different stains, like a Weigert stain, which will emphasize the processes, the axons. But what you can really see is that there are, here I say in layer 1, it's actually very few cell bodies, mostly axons, right? Whereas you see these really big cell bodies in layers 4 and 5, for example. And that's, you have this 6 layers consistent across all of the neocortex, all of the cerebral cortex. Okay, so while all of the neocortex is 6-layered, of course it's different. So neurons in different places will have different thicknesses. So if you go to motor cortex, for example, you'll find that layer 5 is really thick. I'll explain why in a minute. If you go to primary visual cortex, layer 4 is really thick. And based on these differences in thickness, anatomists hundreds of years ago, 100 years ago, went through every part of cortex and tried to figure out where the thickness of these layers changed and gave them all numbers. So when you hear area 7A of parietal cortex, for example, that area is given that number based on its thickness, right? So a lot of, they're called Brodmann's areas. So when you look at a brain, very often all the cortex will have little numbers on it. That's based on the mapping of the layers and where it looks like they're changing their thickness from one part of the brain to the next. Okay. The other thing I should note is that different layers have different projections, maybe not surprisingly, so you have some sort of consistency in the way the circuit's organized across all the different layers of cortex. So I'll give you an example for visual cortex. So if we look at primary visual cortex, which actually sits really at the base of your head, here at the sort of right at the sort of apex of the occipital lobe. Layer 4 receives inputs from the thalamus. Okay, so thalamus is relaying information up from the retina, so we have retina, thalamus, cortex. So this is the first place in cortex that visual information is received. And these neurons then project to other layers of the visual cortex. Layers 2, 3, and 4B project to other layers of the visual cortex. Layer 5 projects down to the brainstem, so it projects to the pons, right down to that sort of, right, the mesencephalon, rhombencephalon, right? So it's projecting down to the pons. It also projects